I'm sorry that I cannot be here in person because I'm here in Marianna. I was presenting here at Peanut Field Day, but I'll be there tomorrow for Ham Field Day and meet you, some of you folks. So today I'll be talking about cotton nitrate study, which we are doing here in North Florida. Um, so this study started last year. So that last year was the first year for this study. And we had two sites for this study. So one was the West Florida Research and Education Center in Jay, which is near to Pensacola. And you can see a aerial view for our site over there. And so we had 6 different rates of nitrogen. So what we are trying to do over here is like we are trying to update the nitrogen recommendation for Florida. So we do have a lysimeters also here in the study. So if you can see like this white spots in middle of the plots are the lysimeter outlets. So our West Florida Research and Education Center site had a lysimeters in there. So our second site was a North Florida Research and Education Center in Marianna. So the reason that we choose these two sites was like Santa Rosa County and the Jackson County are the counties with the greatest cotton acreage. So, and also most of the acreage in West Florida, which is like Santa Rosa and Escambia County, they are dry land. So our site over there is a dry land production of cotton. And here in North Florida, like in Jackson County, near to Marianna, most of the cotton production is irrigated. So we have an irrigated site over here. So these sites were for 2022. So today I'll be talking all about the 2022 results. And for 2023, so we do have an advisory board for this study. So we meet every 6 months, we show what is the progress and get the feedback from them. So based on the feedback from 2022 year, we had a meeting in February. So based on the feedback, so we made changes to 2023 season. So this is the like design for our study. You can see over here, like we have a 25 feet long plots and 24 feet wide. And there is a 12 feet wide alleyway between the plots because there is a outlet for the lysimeters. And we do have 6 nitrogen treatments. So these different rates, different colors in this figure shows like 4 replications. And here is the like plot. So it's 25 feet, 24 feet wide, 25 feet long. And there, it's an 8-row plot and it's planted at 36 inches, which is a normal planting spacing over here. And for the yield estimation, middle 4 rows were used for yield estimation. So we were taking some descriptive data on the second rows from the border, and middle 4 rows were left undisturbed for the yield estimation. So inputs, we use the cultivar Delta Pine 2038. So the reason we use 2038 is like this is a new technology, ball guard tree. So and it's like we had 1646, which is phasing out, and this is taking up that's 1646. So for the nitrogen rate, we had 6 rates, and we used granular urea with urease inhibitor so that we can lower down the volatilization losses. And producers over here use some kind of inhibitors when they apply urea, or in irrigated conditions, they might use some other sources. So all other nutrients like phosphorus, potassium at both sites were applied according to the recommendations from the water slab. And here is the like a treatment structure. So our total rates, you can find like it range from 0 to 25 pounds with the interval of 45 pounds. And last year it was split into 2 applications. So first application was 18 pounds. So it was at true leaf stage. and it is 33% of the current recommendation. So the current recommendation is 60 pounds of nitrogen. So we were applying 18 pounds of nitrogen at true leaf stage. And second split was at squaring or first square. So remaining nitrogen was applied at that time. So fertilizer application was mostly broadcasting. So we used Gandhi to apply the nitrogen. So this is like the visuals from like first application at our dryland site or rain-fed site at West Florida REC, and second is in Marianna. So for the sampling and data collection, so we did a deep core soil sampling to know the nitrate and in different layers in the soil. So at both locations. And I thanks to I provide my thanks to like NFREC Quincy team who came over here in NFREC Marianna to do the deep core sampling for us. And the post-harvest deep core sampling was also done to see like how the nitrogen was moving in the deeper layers. So we did a plant stand count 3 weeks after planting. We took the NDVI and other parameters, leaf chlorophyll. We did a petiole and leaf sampling starting at first bloom until 5 weeks after first bloom. And we also took the above ground biomass and to know like how much was the carbon and nitrogen uptake in the biomass. And it was done at every 4 weeks. So this is the like, again, that's the same visual, but I put this NDVI for one time point, and you can see that NDVI is lower for the control and it's higher. So redder is the box, lower is the NDVI, greener is the box, it's a higher NDVI. So you can see here, like it's very clear, okay, like how NDVI was differing among different nitrogen rates. So we did have a lysimeters in our study in a dryland site. So those were installed last year, and this is just sample video like how we collect our lysimeter sample, and we go over there bi-weekly, and if there is a heavy rainfall event, we go accordingly and collect the leachate sample and send it to analytical lab in Gainesville for analysis. So for the yield estimation, we use 2-row cotton picker to harvest middle 4 rows. And in this aerial view, you can see like there are the gaps in the rows, and those are from the biomass sampling. So we were taking the biomass sampling every 4 weeks. So you can see those gaps. And also at the end, so since like nitrogen application, nitrogen rates can impact the fiber quality as well. So we microgen small sample, 100 gram of sample from each rate, and we send it for the analysis to the Fiber and Biopolymer Research Institute in Texas Tech. And we were looking for the fiber strength, fiber length, and color, and few other parameters. So jumping to the results, so here are the results for the seed cotton yield. We can see over here, like there was no significant increase after 90 pounds of nitrogen per acre. And this is a rain-fed site, but it was significantly higher as compared to 0 and 45. There was numerical increase, but it was not significantly at 0.05 value. And same trend were for the lint yield for the site, but we also need to know what is the lint turnout. So how much lint are we getting from that seed cotton yield? And as the nitrogen rate was increasing, there was decrease in lint turnout. So what does that mean? So it means like either we are putting more energy to the cotton seeds in there, we are making more seeds, or maybe heavier seeds. So we are planning to do the seed index measurement as well this year to know like what's going on, where that energy is going. And here are the results for the Marianna, which is irrigated site. We can see over here, like the yield at the 225 was significantly higher as compared to 90, 45, and 0, but it was not significantly different as compared to 135 and 180. And similar trend was observed for the lint yield in the Marianna side. So here are the results for the Marianna side for the lint turnout. And they were more clear, like reduction with increase in nitrogen rates in the lint turnout, like it was decreasing. You can see over here, like in 225 pounds, even though like in the previous slide, you can see it was higher, but the lint turnout was significantly lower as compared to all other treatments. So these are the like different time points for the biomass nitrogen uptake. And we can see over here, like at 120 days after planting, it was higher under higher nitrogen rates. We can see over here like very clear trend, like how it was increasing at 82 days after planting. So here are the results for the nitrogen leaching. So we got the lysimeter data back a few weeks back, and we can see over here, like, so on the, so for this graph, so on the x-axis, there are like different nitrogen rates, and on the y-axis, it's nitrogen leached, and the bars shows the how much nitrogen was leaching, while the line on the top shows like how yield was increasing. We can see over here, like after 90 pounds, it's in dryland condition, like there was no increase in the yield, but as we were increasing our nitrogen rates, there was increase in nitrate leaching. So we might be losing more to the environment as we are increasing our nitrogen rates. And we will have clear understanding about it, like after we get, so last year it was kind of, we'll receive lots of rainfall, but this year is a little bit dry for us now. So we will have more clear results over the time, like over 2 years from data we get this year. So for the 2023 trials, as I said, like we get the recommendations from the our advisory board, like what changes do we need to make. So one thing that was like that we got back was the site in Marianna station was that need to be moved to the farmer side, because the soil type here at Marianna was much heavier as compared to what farmers have there on their own farm. So, and looking at that means looking at the sites, what farmers have, and it clearly makes sense. So for that reason, we moved our Marianna site to 2 on-farm sites. Again, we are doing like small plot scale study over there, like same design as we followed this year, but it's on farm. So I would like to thank Mr. Larry Ford for providing land to do this study. And second site is other farmer, Mr. Craig Bishop. So this is totally a beach sand, I would say. And we are encountering different problems in this kind of soil as compared to what we saw last year. And other thing that we made change in Marianna was like we increased our splits of nitrogen application. So based on the recommendation, so we divided our nitrogen into 4 splits. Last year we were having only 2 splits, but over here as it is irrigated, they apply their nitrogen in more splits through the pivot or as a fertigation. So to mimic that, we are also applying nitrogen in more splits. And it does make sense, like looking at the sandy soils, there is a more chances of leaching if we apply the soil, if we apply the nitrogen at single time, like heavier dose. So that's why we preferred on splitting the nitrogen. So this year we are having 4 applications of the nitrogen, 15 pounds at plant, but yeah, at true leaf or at plant, and 50% of the remaining at first squaring, which is the stage which cotton needs nitrogen. Most of the nitrogen is taken by the cotton at that stage. And later was like early bloom and mid bloom. But for the dryland side, we didn't got that recommendation from the producers in that region. So we stick to that same treatments for our dryland side. So this is the problem that we found in sandy soils in Mariana. That's a sulfur deficiency. So as we were increasing the nitrogen rates, so we were somewhere messing up the nitrogen to sulfur ratio, and sulfur is prone to leaching as well. So that's why we saw sulfur deficiency, and it's common with the other farmers as well in the region, but we try to fix it. And I'm here in Mariana. I looked up the plots yesterday, and those are looking a lot better as compared to last week. So we applied gypsum as a source of sulfur in this site, because we were not able to use ammonium sulfate due to it, because it's a nitrogen study. So we use gypsum as a source of sulfur. So this is the nitrogen sulfur ratio. You can see here like how with the increasing nitrogen rates, it was increasing. It is usually recommended that it need to be somewhere from 15 to 18, but in our case, it was like 30 or even 40. Uh, Summarizing it, I would say, like, for the dry land condition, we are looking at somewhere, again, I'm not providing any recommendation saying that, uh, somewhere like 90 pounds of nitrogen, but we will have more clear results from this year data. And for the Mariana side, it should be somewhere from 90 to 135. And in the last, I would like to acknowledge FDAX for providing funding for this study, cropping system crew at Jay, our extension agents, Ethan Carter and Libby Johnson, Dr. Barry Tillman here at Mariana providing support, Dr. Sudeep Sidhu and Lakesh Sharma for providing their support, our farmers, Larry Ford and Craig Bishop for their tremendous support, and FREC Mariana and Quincy crew for helping for the samplings in these locations, and our advisory board members for the for keeping us on track and advising us time to time. And thank you. If you have some questions, I will be happy to answer.